So welcome to video number 2, the light-dependent reaction. In photosynthesis, there are 2 reactions that occur. Remember, in cellular respiration there were 3. In cellular respiration we had glycolysis, Krebs, and electron transport chain. In photosynthesis, there are 2 reactions. The first one is called the light-dependent reaction, and the second one is called the light-independent reaction, also known as the Calvin cycle. We're going to talk about the light-dependent reaction first. In the light-dependent reaction, it requires light. It also requires H2O. In the light-independent reaction, we're going to use our other reactant, which is carbon dioxide. We'll talk about that in a little bit. This is by far the most important picture in photosynthesis and the most difficult to understand. So we're going to go through this picture very, very slowly. It is figure 1014 in your book. Okay, and I'm going to have you guys practice going through all these steps because there are a lot of steps in the light-dependent reaction. It is a little bit tricky and confusing. But just to give you an overview, if you notice below the picture, I wrote a general description of what happens. So, first thing that happens in the light-dependent reaction, and as I said, we'll go through this in more detail, is that light strikes the leaf. Where in the leaf is it going to strike? Chloroplast. Where in the chloroplast is it going to strike? The thylakoid. Where in the thylakoid is it going to strike? Specifically, the chlorophyll molecules. So right next to number 1, light, that little yellow zigzag line, you should see it hitting something green, which kind of looks like a lily pad. Okay, so that green thing that it hits is a chlorophyll molecule. Then next to the word light on the bottom in pink, you should see PS2. PS2 refers to photosystem 2. Within the thylakoid, there are 2 different membranes or collections of pigments that absorb light. One is called photosystem 2. The other one is called photosystem 1. The reason why photosystem 2 happens first is that it was discovered second. So just remember, photosystem 2 is the first set of pigments that are involved. So, in recap, light strikes the thylakoid in photosystem 2. Photosystem 2's pigments, those green little chlorophyll molecules, absorb the light energy. It's going to bounce around to a bunch of different chlorophyll molecules. It's going to excite electrons in each of those. It's going to continue bouncing around, exciting electrons, exciting electrons, until it reaches that main structure that says P680 on it. That is the primary electron acceptor. So it's going to excite another electron within there. And that excited electron, think back to cellular respiration. What do excited electrons want to do? Electrons full of energy want to do? They want to go down an ETC, an electron transport chain. So notice what is labeled step 4 in your picture is an electron transport chain. So that very excited electron, which ultimately was excited by the light from the sun, is going to go down an electron transport chain. As we've seen with electron transport chains in the past, the electron transport chain is going to be used to create ATP. There's going to be H+ ions going through, and it will ultimately create some ATP. So, then what happens next? Well, right now we have some very, very low energy electrons. What are we going to do with them? Well, we're going to re-excite them. So, if you look in photosystem 1, those low energy electrons go to photosystem 1, get re-energized, and then they're going to ultimately be used to form NADPH, which is an energy carrier just like NADH. So, to recap, light strikes the thylakoid. The light energy bounces around. It bounces around within photosystem 2. It continues bouncing around until it hits the P680. At the P680, the primary electron acceptor takes the electrons and puts them in an electron transport chain. In the process of that electron transport chain, it creates some ATP. Those low energy electrons go to photosystem 1. Eventually, it bounces around, gets the P700, and eventually forms NADPH. Now, if you're saying to yourself, that was one of the most confusing things I've ever heard, I'm really, really confused, and I don't think I'll ever be able to learn it, we're going to go through it piece by piece. That was just an overview. Okay, so just remember light, photosystem 2, electron transport chain, photosystem 1, NADPH, and now we're going to fill in all the details. In your notes, I've divided the light-dependent reaction into 4 distinct reactions. There are definitely not 4 distinct reactions according to the AP exam or according to your textbook, but I just think it'll make it a little bit easier for you to understand. So, our first part, or our first step, is that light activates electrons. So, the light enters the photosystem 2 and gets absorbed. The energy bounces around to all of these different chlorophyll molecules, activating electrons in each of them. Eventually, you're going to hit the reaction center complex. Okay, that is that little P680 molecule. The reason why it is P680, if you think back to our discussion of wavelengths, 680 represents the wavelength of light that photosystem absorbs really well. So photosystem 2 absorbs nanometer wavelength of 680 nanometers very, very well. So that explains 680. Back to our discussion. Energy is bouncing around, bouncing around, bouncing around, exciting different electrons, and then eventually gets to the center, and the P680 takes in those electrons and gives it to this primary electron acceptor. Before we go on to the next step, I just want to review with you what actually happens to those electrons. So the electrons are getting excited when they are bouncing around to those different chlorophyll molecules. They are gaining energy. And the important part to remember about these excited electrons is that they are very, very unstable, meaning they can't stay in that excited state for long, which is why they then give off that energy to another chlorophyll molecule. Excited electrons are very, very unstable, which is also why they end up typically in electron carriers like NADH, because they are more stable when they're with something else. When I am thinking about the electrons bouncing around, I have a couple of analogies to help you remember all the different chlorophyll molecules, those green little lily pads that they are bouncing around between. The first analogy I have for you is lily pads. So it is like a frog bouncing from lily pad to lily pad to lily pad. The next analogy I have for you is it's like a moon bounce. So imagine you have all of the AP biology class in a moon bounce. And one person, who is full of energy, is going to jump up and then jump back down. Well, when they hit down again onto the moon bounce, that is going to create a force that causes the person next to them to bounce up, right? So think back to when you've been on a moon bounce. One person hits really hard and you go flying up in the air. That is similar, good analogy to keep in mind for what's happening with these electrons. One electron gets excited, then it comes back down, and then another electron gets excited and comes back down. Another electron gets excited, comes back down. So it's kind of like a moon bounce. If that analogy confuses you more than helps you, then just ignore it, but hopefully it's a little bit helpful. So now we have our electrons present, full of energy, in the primary electron acceptor. That primary electron acceptor is the primary electron acceptor in the electron transport chain. So, what happens next? Those electrons that have a ton of energy are sent into an electron transport chain. Just like in cellular respiration, where the electron transport chain occurs in the inner membrane of the mitochondria, in photosynthesis, the electron transport chain occurs in the thylakoid inner membrane. In that thylakoid inner membrane, the same things happen as happen in the electron transport chain in cell respiration, meaning the electrons slowly give off their energy, there's some cytochromes involved, and when those electrons are given off their energy, they're going to give it to H+. What's the H+ going to do? It's going to move across the membrane. Then that H+ is going to want to come back down, and it's going to form ATP by going through the ATP synthase. So, we have an electron transport chain being used to create some ATP during photosynthesis in the light-dependent reaction. Also, when those electrons are given up to the primary electron acceptor and going through the electron transport chain, that means that the P680, who ultimately gave those electrons, donated it to the primary electron acceptor, is now a positively charged molecule. It is very, very, very unstable. So as a result, the water comes in. Okay, if you look at that little red circle on the side, and then the little blue H2O molecule, the H2O is going to donate its electrons to the P680. Why is that important? Why do we care? Well, it makes it stable. In addition, it shows you that why water is needed in photosynthesis, and if you look at that little red circle, it forms oxygen. So the reason why there is oxygen in the atmosphere, the reason why anything that breathes aerobically can exist, is because in the light-dependent reaction in photosynthesis, water is split apart to give electrons to P680, and a byproduct of oxygen is formed that is then given off by the plant. Final thing about this picture that is important is the term photophosphorylation. What does phosphorylation mean? To give a phosphate to something. So, what does photo mean? Occurs in the presence of light. All photophosphorylation means, sounds like a very big term, sounds very confusing, just means ATP is formed using light energy. So ultimately, what provided the energy to form the ATP? Well, how did the electrons get excited? Light energy from the sun. Photophosphorylation, just phosphorylation, forming ATP molecules using energy from the sun as its light source. So now at this point we've formed some ATP, but our electrons are very, very low in energy. They have gone down the electron transport chain. So what happens next is that light energy, as you can see on the very far right, is going to hit photosystem 1. That then ultimately is going to excite those low energy electrons so that they can have energy again. So light energy hits photosystem 1, bounces around to all those different chlorophyll molecules. Ultimately, the energy reaches P700, which is the wavelength of light that is absorbed best by photosystem 1. Those low energy electrons get excited again and they go to the primary electron acceptor. The same basic process that happens in photosystem 2 happens in photosystem 1. So once again we have these very high energy electrons at the primary electron acceptor in photosystem 1. What happens, as you can see in step number 7, is another electron transport chain. But this electron transport chain is a little bit different than the electron transport chain we saw in photosystem 2, and also the electron transport chain we saw in cellular respiration. Notice, on the right side of the picture, there's no ATP made. So what happens to that energy? Well, if you look at the little electrons, they go to form some NADPH. So this electron transport chain's function is not to produce ATP. It instead is to store energy in the form of NADPH. NADPH has the same function as NADH. So the function of NADPH is to store the electron's energy for use in a later reaction. As we're going to see, the NADPH is used in the light-independent reaction known as the Calvin cycle. The way that you can remember NADPH is in photosynthesis is that it has a P in it. P technically stands for phosphate, but if you could just remember that the P in NADPH stands for photosynthesis, then you always remember that NADPH is used in photosynthesis and NADH is used in cellular respiration. Also notice, there is a molecule called FD in the electron transport chain. That molecule is known as ferredoxin. It is mentioned and brought up on some of the AP exams. Just know that ferredoxin is an iron-based molecule and that is used in the electron transport chain in the light independent reaction.